As we talk about reproduction and adaptation in prokaryotic cells, one very important thing to point out is that they are going to be reproducing asexually. So the process that we're talking about here is specifically called binary fission. And binary fission is a fairly simple process where the chromosome is going to be copied. So we'll have the DNA copied, and then the cells are going to split into two. So the DNA is copied and the cell splits. When that happens, each cell will now have a copy of the circular large chromosome, and this is also going to produce exact copies, or what we would call clones. Okay, so what we get from all of this is something that should be exactly the same as that original cell that we started with. When we're talking about binary fission, this is going to be a very fast process. So by very fast, this can be as fast as 20 minutes under ideal conditions. It's not usually 20 minutes though. So typically we're not talking about ideal conditions. We might be talking about an hour to 3 hours, or if we're talking about E. coli, which is a typical bacteria that we find inside the human body, under normal everyday conditions in our body, we're really looking at something more like 24 hours. Okay, but if you take those same E. coli cells and you put them in a lab and you give them ideal conditions, they can do this replication in 20 minutes. So prokaryotic cells, through this process, are able to reproduce exponentially at very fast rates. This is one of the reasons why prokaryotic cells are so abundant on the Earth's surface. Now, another thing that leads to their abundance on the Earth's surface is because they have abilities to withstand very harsh conditions. And an endospore, which is what we're seeing on this slide, is one of the ways that they do this. So with an endospore, what's going to happen is the cell copies its DNA, and then it takes that copy of DNA and it basically packages it in a tough container. And that very tough container is the endospore. That endospore is then going to be dehydrated, and then the cell destroys itself. So after it has the DNA copied, packaged up, and dehydrated in this nice little container called an endospore, the cell will lyse. And when we say that the cell lyses, that's the same as saying that it's going to burst. So all of this, which is the remainder of the cell, is going to go away, and all we're left with is this nice little endospore in the middle. This endospore is very hardy. So this is something that can survive for years. It could survive for hundreds or thousands of years in very harsh conditions. So if there was a time when there were very harsh conditions, a bacterial cell could produce one of these endospores, and that endospore could lie dormant for a very long period of time. And then when conditions become favorable again, that endospore could develop into the normal bacteria. So this is going to enable cells to withstand some very harsh conditions and environments. So all in all, prokaryotes are the most abundant organisms that we have on the Earth's surface. There's a few different reasons for this big abundance of prokaryotic cells, and we've touched on some of these ideas already. One of them is the fact that they have a very rapid rate of reproduction. They can reproduce exponentially, and we said that those generation times can be quite short. Another reason is mutation. Mutation is a change in the DNA. And then we can also get some genetic recombination. So when you take all of these three things and you package them together, this is going to lead to organisms that are able to adapt very rapidly to new environments. So if we talk about the rapid reproduction and mutation to start with, prokaryotic cells are going to reproduce asexually. And we said the process is going to be binary fission. This should produce clones, which are exact copies. There are chances to get errors anytime the DNA is copied. And if we have these generation rates that are 20 minutes or an hour or a day for that matter, we are producing a lot of new bacteria. So if we look at the error rate that we typically have, the error rate is quite low when we're talking about prokaryotic cells when they copy their DNA. So the error rate is 1 in every 10 million bases copied. So by error rate and taking place during the DNA replication process, this could be a base pair substitution, it could be an insertion, it could be a deletion. Okay, but we do have some errors that are happening. May not sound like very much when we're talking about 1 error in every 10 million, but if you look at this statistic here, it's quite staggering. So with 2 times 10 to the 10 new E. coli cells per day in a human's intestines, so we're just talking about in a human's intestines that every day we are going to have this many new E. coli cells. When you consider that and you consider the size of the genome in those cells, that's approximately 2,000 newly mutated bacteria per day. So 2,000 newly mutated bacteria, that's a lot of changes. So even though we have a very low mutation rate, when you take into account the quantity of bacterial cells and how fast they're reproducing, we are getting quite a lot of mutations. And that's going to be the raw, if you remember, when we're talking about new mutations, this is new genetic variation. And so we say that bacteria reproduce asexually, and that should be producing clones, but that doesn't mean that we don't have a mechanism of getting some genetic variation. There's quite a lot of genetic variation coming from this mutation process, and new genetic variation is the basis of natural selection. Okay, so let's not forget about the fact that that is what natural selection actually operates and works with. Genetic recombination is another way that we can get some genetic variation inside prokaryotic cells. The name here, genetic recombination, means new combinations of DNA. And so this means that we're going to be piecing together pieces of DNA so that we get a new chromosome compared to what we had before, or there will be new DNA inside that cell compared to what we had before. This is going to be responsible for what we call horizontal gene transfer. And horizontal gene transfer is extremely important when we talk about prokaryotic cells because this is transfer of DNA between two species. So just because we're not reproducing sexually in prokaryotic cells, it doesn't mean that you can't have DNA swap between two species. So we're going to look at how that can happen. We're going to talk about 3 mechanisms for genetic recombination. We have transformation, transduction, and then we also have conjugation. So the first one we're going to talk about is transformation. And with transformation, this is very simply the uptake of external DNA. So it's the uptake of external DNA, and then that DNA is going to be incorporated. And by incorporated, it's incorporated into the chromosome. So if we look at this in a picture, we've got some DNA fragments that are outside of a prokaryotic cell. When those DNA fragments come into contact with the cell, some cells have the ability to take that DNA inside. So they import the DNA into their cytoplasm. Once that DNA is inside the cytoplasm, the prokaryotic cell is able to use the process of recombination, which means it's going to swap out the new DNA for some of the pieces that it has on its own chromosome. So if we look closer at this picture, the bacterial chromosome has an allele lowercase b. What it can do is it can swap this version of B, which it just uptook from the environment. So it can swap the versions of B so that in the end, we have this new chromosome compared to what we had before. This is very similar to the process of crossing over that we have in eukaryotic meiosis. The new chromosomes are called recombinant because they have a new combination of genes or a new combination of DNA compared to what they had originally. So transformation is something that can certainly contribute to genetic diversity when we're talking about prokaryotic cells. Another mechanism for this is what we call transduction. And with transduction, we have the involvement of phages, which are little viruses of bacteria, and these are going to carry the DNA to a host cell. So now we have an organism or a virus that's actually bringing the DNA into the host. Okay, so if we look at this process in this picture here, we really have two cells involved because we're going to have a donor cell that's going to donate DNA to the phage, and then the phage is going to take that donated DNA and it's going to transfer it to a host cell. So in the first part of this picture, we're seeing the phage infecting a donor cell and it's going to be collecting that DNA. So here's our donor cell. The phage is going to be synthesized inside the donor cell, and when it does that, note that it is possible for the phage to pick up some of the DNA from the donor. So now it has new DNA inside of it. This phage can then go and infect another cell, which is going to be the recipient cell or the host cell, and it injects that DNA. And then just as we saw with the transformation process, it is possible for recombination to take place. And with recombination here, we are swapping out versions of the gene. So in this one, the cell is going to swap out the A- for the A+ version of the gene so that we do end up with a cell that now has a new combination of the DNA on the chromosome compared to what it had originally. So with transduction, we have a phage that is mediating the transfer of DNA from a donor bacterial cell to a host or recipient bacterial cell. The third type of genetic recombination that we want to talk about is conjugation. And with conjugation, we have DNA transfer between bacterial cells. So this will be a direct transfer from one cell to another. This is going to be a one-way transfer of DNA. And that means that one cell will be the donor, and that donor will contribute its DNA to a recipient cell. If we look at this process, here we can see two cells that are now connected together with what we refer to as a sex pilus or pilus. We have one cell that's going to be the donor. The donor is the one that actually forms the sex pilus, and it then attaches it to the cell that's going to be the recipient. So over here, we have our recipient cell. The pilus gets attached, and then it will actually draw those two cells closer together so that they're in close proximity. Now, at that point, sometimes the DNA is passed directly through the pilus from the donor to the host cell, to the recipient cell. Sometimes we get what's called a mating bridge that's formed, and that will be the little channel or tunnel that the DNA is going to pass from. But we will be taking DNA from here, and we're going to pass it through a tunnel into the recipient cell. So this is a transfer of DNA. Now, the ability to do this is not something that all bacterial cells have. So not all cells are able to form the pilus and transfer the DNA. There is what's called the F factor. and the F factor can be held on a plasmid. This can be something that's on the chromosome itself. This contains the genes that are necessary for forming the pilus and transferring the DNA from one to the other. So if a cell has the F factor, then it's going to be able to serve as a donor cell. If we look at an example of the F factor on a plasmid, that's what we have right here. If the F factor is on a plasmid, we call that plasmid the F plasmid. And this would be what we refer to as an F positive cell. So an F positive cell has the capability of serving as a donor cell. What's going to happen is the F cell will form that pilus and then the mating bridge between the two. Once we have the mating bridge formed, now we have the tunnel to actually get the DNA transferred from one cell to another. The plasmid will be copied, so we'll have a slight little cut in the plasmid so that we can begin to copy it. And then notice that that plasmid DNA is going to be transferred from the donor cell into the recipient cell. And as it's transferred, we go ahead and we make the replacement copy in the F positive cell, and we're also going to get the DNA copied as it goes into the new cell. So when all is said and done, we have gotten an exact copy of that plasmid transferred to the new recipient cell. If this is the F plasmid that we're transferring, now that recipient cell then becomes competent of also performing this conjugation process. So that one is able to now attach to another cell and transfer DNA into it. We can also have the F factor carried on the bacterial chromosome itself. If this is the case, it's a very similar process to what we had in the last slide. We will have attachment of that donor cell to a recipient cell. We'll get the pilus formed, we'll get the mating bridge formed, and then we will have a small cut in the DNA, and then that DNA will be transferred through the mating bridge into the new recipient cell. When it's transferred, we will have now a piece of DNA inside the recipient cell, and now it's a matter of recombination. As we saw with transformation and transduction, we're going to swap out the new genes for the old genes that we had in that cell. So when all is said and done, we have recombinant DNA or recombinant chromosome in this new recipient cell because it now has a new combination of the genes on the bacterial chromosome. You've probably heard about antibiotic resistance with bacteria or with prokaryotic cells. And this is something that is very important and plays a major role in medical treatments. Because we have so much gene transfer between bacteria cells, if one becomes resistant to bacteria, that resistance gene can easily move into another bacterial cell. So antibiotic resistance genes can do a number of different things. They can be genes that produce proteins that are capable of degrading antibiotics so that when you treat the cells with an antibiotic, they're able to break that down before it causes any damage. We can have ones that are able to alter the antibiotic to some extent so that it's not as effective as it was before. In this picture, you're seeing it actually coating the surface of the antibiotic so that that antibiotic cannot affect and target the proper enzymes and proteins inside the bacterial cell. And we can have ones that will actually make a transport system and get that antibiotic right back out of the bacterial cell. So a lot of different approaches here when we're talking about resistance genes in bacteria. There is, though, keep in mind, lots of gene transfer. So if we have lots of gene transfer between bacterial cells, it's very easy for new species of bacteria to gain an antibiotic resistance gene. Now, if we just look at how the overuse of antibiotics can affect antibiotic resistance bacteria or lead to antibiotic resistance bacteria being prevalent in the environment, let's look at this picture. We can have a normal population of bacteria that has a lot of genetic variation there. We've already talked about ways that you get that genetic variation. So this might just be the result of mutation. There could have been some recombination events that took place as well. But when you take that varying bacterial population and you apply an antibiotic, what that antibiotic is going to do is it's going to kill off sensitive bacteria. And that's exactly what we have in mind when we apply antibiotics. Now, where the harm comes from is that you are very likely to have a few prokaryotes there, or bacterial cells, that already have an inherent resistance to that particular antibiotic. So they're going to have a high resistance level, which is what we see in this picture. Since you have weeded out and killed off all of the sensitive bacteria, now the ones that are left to reproduce new bacterial cells are the ones that are resistant. So we end up with a population at the end which is genetically related to the ones that were very resistant. So we have a population that's very resistant to antibiotics. This happens if we're using antibiotics. Now, antibiotics serve a purpose, but we don't want to use them unless they're really necessary, because we run the risk of turning that varying population from a range of antibiotic susceptible bacteria into a population that's very resistant to the antibiotics. A lot of times, doctors might use an antibiotic cocktail, which means that we have a mixture of antibiotics. And with the mixture, we're increasing the probability that we don't have bacteria there that are going to be highly resistant to multiple antibiotics at the same time. So this is just another approach to try to not only kill off bacteria, but also to stop the production of these very antibiotic resistant populations.